I would like to introduce the first speaker, which is a very dear friend of me. I know him more than 20 years ago, Dr. Noureddine LOUANJLI, who is a very active Vice President General and Executive Director of the Association for Scientific Research and the Reproductive Medicine Endometriosis Research Group and IRI Center Fertility at Casablanca. Dr. LOUANJLI was graduated from medicine at the University of Strasbourg, Department of Clinical Biology, Alsace, and he is the head of Laboratory for Clinical Analysis and Assisted Reproductive Technology, IRI Center Fertility, African Center, Casablanca, Morocco, and is a member of MSERM and ISRI, and he has done a lot of scientific, interesting research, and is founder of the Moroccan Society of Reproductive Medicine and founder of the actual society that invite and guest this meeting. Dr. LOUANJLI, please start your lecture.
Yes, thank you. Thank you very much. You have finished?
Yes, please.
Thank you very much, Professor Amer, and I hope welcome to all attendees and speakers of this first event of our association.
Your title will be Impact of pH Media.
Yes, exactly. You see my slides?
Yes, very well.
Okay.
Yes.
I have problem. Yes, exactly. What problem? Okay. Okay, thank you. Our topics today. One second, please. Okay. Our topics today is the study of important parameters in embryo culture that influence the results and the quality of our work. It's well known that during in vitro culture, many stress can affect oocyte and embryo competence, especially competence can have devastating effect. We have osmolality, light, toxin, pollutant, mechanical effects, temperature changes, chemical product, pH, it's our topic today, and oxidant if we don't work at the right oxygen tent. The major intracellular processes are highly sensitive to pH changes, including protein synthesis, enzyme activity, oxidative metabolism, mitochondrial function, cytoskeletal regulation, and energy production. In consequences, intracellular pH regulation is a vital aspect of cell homeostasis. We have a lot of cell membrane contain intracellular regulatory mechanism. There are a lot of exchanger that located in cell membrane of oocyte. It's well known that oocyte cytoplasm has limited buffering capacity to regulate its own pH, and pH intracellular follow the pH medium that has to be well calibrated. It is influenced by protein, amino acid, and other media components. As you see here, there are consequences of pH on the cytoskeleton that regulate positioning on the cell spindle and chromosome alignment. And also pH contribute to the distribution of the mitochondria, which is correlated of the development competence of the oocyte and embryo. Lowering pH decrease glycolytic activity and increasing pH influence oxidative metabolism. The metabolism is severely affected by the huge changes of pH medium. Swain in 2014 have demonstrated the mean oocyte intracellular pH values at about 7.0 in human. Ovarian oocytes that are growing and have to achieve their fully grow in the ovary lack the ability to regulate pH intracellular and need cumulus cells around to regulate need these cells to regulate to the pH. Granular cells support pH regulation during oocyte growth, and maturation. This support is lost with the denudation process for ICSI procedure, and handling for ICSI is processed the day of ICSI. We remove all the cell cumulus from the oocyte, and he is vulnerable to pH changes. And you see here that during early stages of embryo development, the ability to maintain normal cellular functions such as regulation of intracellular pH, relief of oxidative stress, and ionic homeostasis grow gradually. And the oocyte is very vulnerable, and the blastocyst is present relative resistance to changes. As we know, the pH in the medium level is achieved through a balance between CO2 level of the incubator and the bicarbonate in the medium. It depends on the incubator and the medium types. We reach the target working pH media for oocyte and embryo culture after certain hours. It depends on dish will cover or not of oil of volume and density of this oil, medium droplet, volume, starting pH, type of incubator. At least we expect 8 hours at least to equilibrate the medium for working for IVF. It's the very known the curve to stabilize the pH. It means we have to prepare the lab 1 day before to have optimum condition of pH value for working at the day of ICSI. Factors that could affect pH changes during embryo culture. We have temperature. Embryologists that want maximum accuracy will opt for a pH meter that come with a temperature compensator probe, because all our measures are made at 37 degrees. We need the probe that compensates this temperature. Made caution, be careful. The handling medium have buffered by HEPAs and MOPs have changes in pH at different temperature. If we have to check this pH before working at 37 condition, and we have an example with the 37 condition. The MOPs could lower his pH, and it could be deleterious for the oocyte. The second condition, the production of ammonium, it's second condition that could change pH during culture. Embryo development, differentiation in culture, embryo endometrium, pH regulation, and gene expression in prolonged culture are affected by modification of pH and production of this ammonium. However, our current product medium now are replaced glutamine by dipeptide form that generate less ammonium, and there are less changes in pH. pH stability depends equally on osmolarity, and we have to use a right oil to overlay the droplet, culture droplet. And the Swain in 2018 demonstrates that the heavy oils stabilize the pH and osmolarity, and it fitted with the new generation and of incubator that we keep embryo for 5 or 6 days. Heavy oil, better stability and osmolarity in pH. We have here manufacturers that produce different density of oils, or can have all density in the commerce, in the market. Embryo metabolism change during early development stages. We have what is the right value. Here, Swain 2002 demonstrate that the better oocyte, the better value for oocyte, it's 735, between 730 and 740. We have the maximum fertilization rate at this pH. And after at the cleavage stage, Karen Miller 2014 demonstrated that between 7.2 and 7.3, we have the higher maximum cleavage rate and the maximum quality of embryo at this stage. And the same author demonstrates at 7 for blastocyst stage, we need between 730 and 740, a mean value of 735. This finding doesn't fit with our practice daily, because the new generation of incubator needs a stable condition, one condition for 5 or 6 days. And the embryologist decide for the pH value according to his own experience, 730 or 730, 25 to 27, to not to modify, because we can, the target of this new generation of incubator to make homeostasis stable and not to modify product and incubator, and we have to choose one value of pH during all culture. Here, the value that are recommended by the producer, the firm of the companies, it's in general between 7.2 and 7.4. But every company you have little changes in according to the concentration of bicarbonate between companies. Here, for example, we have the same company and the same medium, but the two lots. If you have one lot works at 7% CO2 and other lot we must have 7.5 for the same target pH, 8.3. We have to check at every new lot. We have to check the pH, even for different company at the same basal buffer. In the right, you see here for HTF basic medium, you have a different pH in function of the company. You have to try to check your pH, even if for the handling medium based EPS or MOPS. Here, equally, it depends. The value of pH depends on the incubator and the media. You have if you work with an incubator, you have a target value of 7.3 for pH. If you change the incubator, you have to recalibrate with the new incubator. It's the value, for example, in this condition with the same value, we need 6 for the first incubator, 5.5 for the same incubator, and we need perhaps 7 for the third incubator for the same pH target. We have to calibrate it according to a new incubator for the same medium. I conclude that the optimal pH use will be according to the media, the incubator, the culture conditions, the lab experience, and embryology goal of each laboratory. It's the embryologist in his lab that decide which value he keep for all culture condition. A few words for measurement. We have classical pH meter. It's abundant now because it consumes a lot of volume and it's quick. It's not quick and not adapted for AFR laboratory. Now we have the probe inside the incubator with lecture on live lecture. We work and you read what happens in the incubator. Our choice is we chose this kind for blood gas analyzer. It need less than 80 microliter auto calibration once per lot and adapted to culture under oil. Result in 5 minutes, and you can check quickly multiple incubator at the same day. We have the use in our lab. It takes less than 30 seconds to have the pH of your working medium. And if you have the value and you can check your incubator, it takes 10 seconds. It's our choice. It's a gas blood analyzer. The accuracy of any pH meter should be validated prior to implementation in clinical AFR laboratory. Thank you.